Morning. I'm going to get started and hopefully be closer to on time or even early today because I actually have a plane to catch, unfortunately. So what we talked about on Wednesday, and I'm going to mirror again today, is really this relationship between thalamus and cortex. And I tried to hit some of the big overview points about it. General spatial organization of cortex, and similarly general spatial organization of thalamus, and trying to get you to sort of be able to visualize the extent to which they align, so that as much as you can remember one of them, it gives you a crutch to remember the other. The other is to think about what they do and how they interact, and that thalamus in some parts is really a direct relay. It's one more step in a sort of multineuronal synaptic chain that goes from a sensory modality, that sensory relay, where it's really functioning in many systems sort of as the third order neuron, to the fourth order neuron, which is in the first primary somatosensory, primary visual, primary auditory cortex. Okay? And then the second is on the motor system, the fact that the motor system feeds back on itself continually, so that we have our motor cortex projecting not just to the spinal cord, but we have it projecting to the basis pontis, through the basis pontis into the cerebellum, where it meets other types of information, and then back out of cerebellum to both red nucleus to descend both to the cord and back into another feedback loop to the olive, as well as to the thalamus, and that thalamus talks to cortex. Okay? And the basic other idea that I wanted people to get was that, in some sense, when you think on the sensory side, and I'm going to make that slightly more posterior because somatosensory is posterior to motor, you begin with the sort of simplest. That's the areas of visual cortex that respond to moving angled lines of light. Okay? That's about as far away as you can get from your visual sense of the world. And that gradually those build to more and more complex within that modality. So eventually we add in contrast, depth, color, and sort of create three-dimensional objects. And then those are going to go in parallel and join from one modality into multimodal. Okay? So you go from the incredibly simplistic, intrinsically hard to understand how you get from there to your sense of the world, to the complex picture of the world. In motor output, you do the exact opposite. You begin with the most complex. I have this opportunity. I'm trying to make a decision about it. Should I do it now? Should I answer the phone? Should I not answer the phone? Do I need to do this? Do I need to do that? What's most important now? The most complex. And you work to lower and lower levels of complexity until you're finally down to the, let's just drive anterior horn cells in the spinal cord for the biceps on the right and inhibit those for the triceps on the left. And we'll do that for 12 milliseconds, and then we'll start using brachioradialis, and you make a movement. So for motor output, you begin from the most incomprehensible complex and go to the simplest output. So they are essentially oriented in opposite directions. You can think of this, the motor, where's my point? Where's my little, here I am. You can think of motor as being from the broadest coming down to the narrowest, and sensory as being from the narrowest and going out to the broadest. Okay? So that organization. But all of that decision-making isn't based on just pure cortical information. It's going to be modulated. And it's going to be modulated by thalamic connections. So those multimodal association areas will have different parts of thalamus. And we're going to think and assume that they function sort of like a sensory thalamus. How come? Well, because it's really the only model we have. Okay? And in fact, all of the conceptual structure of this really is built off the visual system. And visual system turns out to have been the easiest one, although it was very complex, but the easiest one to manipulate and understand, because you could figure out what a stimulus was. Okay? If you think about somatosensory stimuli, how would you figure out exactly what to do? Okay? Well, all of that sensation is a mixture of multiple modalities. Okay? If you think about auditory, pure tones probably could have done pure tones, but animals, but they didn't. Smell is really complex. Taste is almost impossible to think about. But vision, you figured you had to shine a light in the animal's eye and record. And by changing what you did with the light, you could eventually figure out, and John Assad will show you a wonderful video of Hubel and Wiesel actually recording and showing you how they did it. If you think about the way people do experiments now, you will discover that it is incredibly primitive. It's basically with a projector with slides on a basically screen that's in front of the cat, or actually the monkey sitting in the chair. And they're just doing it, and as they mark things, you know, one of them goes up to the board and scribbles something down. Talk about data acquisition. But vision you can understand. You know what the stimulus has to be. And then you can see how neurons at each level, how do neurons in the retina respond? How do neurons in the lateral geniculate respond? How do neurons in primary visual cortex respond? And you can see that they are driven by different eyes. They're driven by orientation. And then you can move to the other associational visual cortical areas and say, what does it look like? And so all of our sort of concept of how you're going to think about thalamus to cortex is essentially the extension of that model. And there's no reason to assume that especially the non-sensory relay pieces of thalamus work the same. But it's really hard to think about how you design an experiment to be able to say, what is it about patterns of activity that drive it? What are the equivalents of ocular dominance columns when you're talking about motivation control? So there's an enormous amount there that we still don't know. It's what my PhD advisor, when central CNS synaptic physiology was really poorly understood when I was a graduate student. And everything was assumed to behave just like the neuromuscular junction, okay? Because everybody knew how the neuromuscular junction worked. And it's what my PhD advisor called the meathead theory of the brain, okay? It was the only model you had, so the assumption was everything was going to be just like it. And it turns out most CNS synapses, many CNS synapses are completely they're not at all like neuromuscular junctions. The methods of inactivation are completely different. The sort of anatomic implications of them are different, but it was the only working model that was had. And the visual system is really the only operational model. So when we think about the modulation, we're going to have to think about both combined modalities, the so-called multimodal, and I need to put these in the other order, but also things that basically tell cortex to be doing. And when we talk on Wednesday, we will talk about cortical networks. And those cortical networks you switch between them. The ability to pay attention, the ability to not pay attention, the ability to think about language, the ability to think about spatial. All of those are alternative clusters of cortex that are wired together, that function together. And they are driven to be, which one is on at a moment is in part controlled by thalamus. So again, we think about our cortex. We have our maps that let us know where we are. Homotypic is what sort of cortex? Oop. Oh, I didn't have the labels. Sorry. Yeah, so this is going to be our association cortex. And it was thought to be the sort of dominant pattern because you have far more of that than anything else. Granular is sensory. And that's because the big pyramidal cells that make up agranular are your motor output neurons. So that is your motor cortices. When we think about anatomic distribution, a tiny amount of brain is actually devoted to unimodal sensory or primary motor cortex. So the blue here, what gyrus? Right, so that is our post-central. And what lobe? Parietal. It is the anterior-most boundary of the parietal. If that's the anterior-most boundary of the parietal, what's the pink? What modality? Yeah, it's motor cortex. It is the gyrus? Precentral. And it is what lobe? Yeah, it's the posterior boundary of the frontal lobe. Okay? This little blue dot here that's on the posterior portion of the superior temporal gyrus. Oh, I gave away the answer here. So that is posterior, superior, temporal gyrus. And that is We've got somatosensory. We've got motor. Where's visual? Not there. Exactly. Okay, so what do you got left? Auditory. So that's your auditory primary space. It's tiny. It actually is almost invisible from the surface. It's mostly running horizontally in here. And then back here on the medial surface, you've got, this is, yeah, so it's Brodmann area 17, also known as striate cortex. Why is it striated? The line of Gennari, okay? You can see that line. That's why it was called striate. And that's our primary visual cortex. What have we got? What is it? Thalamus. Okay, is it a left or right thalamus? It's a left thalamus. What are the bunch of clues apart from the fact that it's labeled sort of up at the top? Yeah, so if you look at the two geniculates, the lateral geniculates are at the anterior or the posterior boundary of the thalamus. Posterior. All right, so that tells you that this is back. And this is anterior. The other clue is that the anterior nucleus is here. And the posterior nucleus, is this the posterior nucleus? No, it's P4 pulvinar. So that's your anterior posterior. And then you look at your geniculates and you go, okay, so this is medial. That must be the midline. It has to be a left thalamus. Okay? And as you look at it, you've got your anterior grouping together VA, VL. What is the functional component? Is motor. VPL, and where's the next part of VPL? VPL pairs with VPM. Why can't I see VPM? Because it's deep, it's medial, it's in closer in. Okay, and they are somatosensory, lateral, function, medial, auditory. All right. And then the others we're going to talk about over the course of the morning. So as we line them up, you can color code them, and there is this spatial relationship. Anterior and cortex and anterior thalamus, posterior cortex, posterior thalamus. It's a relatively stable, relatively consistent across the whole set of structures. And when you lie it out and do this sort of MIP projection equivalent, they actually do map. If you look at this figure, you will discover that it comes from a book that uses the full formal Latin names for everything. Okay? Pulvinar thalami pars anterior. Don't worry about them. So when we think about these thalamic relay nuclei, they're inputs. They bring in sensory information. They actually get information from the cerebellum and basal ganglia, and we'll see that. But they also are reciprocally connected with their cortex. And although that's there, we're not going to talk about it much more at all. And the outputs go where? Mostly to layer 4 for sensory cortex. That's the line of Gennari in the visual cortex. That's the terminal field of the geniculate projections. Okay? And then for the others, we go up and above and below. All right. The associational areas, they are mostly hearing from cortex. They are mostly about regulating cortex, so they're going to mostly be innervated by cortex. And their output is back. So it is a reciprocal. This is a, thalamus, cortex submultimodal, okay? Back and forth, and they are going in up in layer 1. What's in layer 1? What type of cells? Granular, pyramidal? None, it's molecular layer. What is in the molecular layer? Molecules is not the right answer. What? Well, it's got the axons here, and it's got dendrites of all those, the apical dendrites reaching all the way up there. So that's what we've got up there. All right? So just to remind people, we looked at the visual system. We have our optic nerves, chiasm, tracts, and radiation. We have the loop of the radiation that extends into the temporal lobe, known as Meyer's loop, which carries superior visual quadrant, okay? Because on this side, this is only one side of space. So it's superior visual quadrant, and as you'll see, superior quadrant goes to the inferior bank of the visual cortex. Okay? So we've got that sweep. So a lesion in the geniculate will give you what? Lesion here. So this is which geniculate is this? Left or right? Everybody's tipping their head, rolling around. Is it left or right? I love watching the head movements. Okay? It's left. Because you're looking from the base of the brain, so in order to see what it is, you have to roll it over. Okay? You know it's the base of the brain because you're looking at the cut sort of midbrain here. It's actually cut right at the posterior commissure level, right there. What's running on the posterior commissure? Fibers that came in like this, synapsed here, and they project here and here. Well, they're not Edinger-Westphal. Edinger-Westphal is the target, but they're the crossing fibers of the pupillary reflex crossing that posterior commissure there. All right? So if you lose this, which is the left lateral geniculate, what do you lose? If I think about my visual fields, I lose which field? Contralateral, which eye? Both. So I lose the right eye, the right field in both eyes. Okay. So now I didn't make this very clear on Wednesday, so I wanted to sort of touch on it again. So when we think about that visual cortex, and we have to organize the way space is represented, okay? We've kept space as a map. The retina has a map of space based on where light is hitting it from, okay? That map is dependent on the inversion from the lens, but don't worry about it. We care about space, okay? When space comes in, it's maintained in the geniculate as well as in the colliculus. That collicular map lets us move eyes. So we've got a coordinate map there, and we've got a coordinate map of where information is coming in the geniculate. And we maintain that coordinate map when we get to the visual cortex. And here's the orientation of it. The center of so if this is my visual field in this eye. So let's just draw this eye. And the visual field for this eye, this is the part of space that I'm worrying about right now, okay? I'm looking at the contralateral eye, okay? And so I'm worrying about this part of space. Right here at the center of vision at the horizon is about as close as you can get, where's my pointer, to the occipital pole. As you move along this sulcus here, you move from the posterior cortex more anterior, you move from this center of your visual field out to the periphery. If you go superior in the cortex, where's my pointer again? If you go superior in the cortex, you go down in space. And as you go inferior in the cortex, shown here by the slightly blue lines, you go up in space. Okay? So at the very, very back of the visual field is the center of vision. As you move anterior, you go peripherally, and then they are inverted. Superior bank is the lower half. Inferior bank is the superior half of that quadrant of that half of space. Okay? Now, what happens if you fall on the back of your head and you bruise the occipital pole? What part of visual cortex might you damage? Okay, so you bang your head, you smack and you damage back here. And what sort of visual field deficit do you get? You get a central one, okay? All right. And we'll see various examples of this and the patterns of how these are preserved and where they sit when we talk about stroke on Wednesday. Next Wednesday, we will cover the cortical vasculature as well. All right. So when we think about visual processing, and the reason I'm using vision again is because we know far more about it than any of the other modalities. Okay? We really have tools to understand it, unlike everything else. So everything else is done by inference and sort of simple examples, which is as you go around this, and you'll see on Wednesday that there are in fact two distinct pathways. One of which goes from first order to second order and higher order processing moving towards the parietal lobe. And there's similarly processing that goes on moving towards the posterior temporal lobe. And there are different types of visual processing that go in either direction. But in general, you're going from very simple response to moving lines to complex processes. And there are lots of issues about where exactly is depth perception, for example. It seems as though depth perception mostly happens sort of someplace out here on the lateral aspect. Where are color perceptions and things like that? And that color perception turns out to happen more down in here. But we have ways of trying to get at that both from people with lesions and people without. But you get this basic idea that you're going from a sort of central core to surrounding rings that have got higher order processing. Your information is being manipulated in a way to give you a more complex set of data. So now here's a figure that should give you lots of sense of familiarity. Okay? We've seen this before, except I cut it off, right? I cut the top part of it off from about, my previous one, I showed this before, I didn't show you this. Okay? So what have we got? Actually, no, I didn't show you this. I cut it off here, right here. So what are these two nuclei here? Yeah, the two cochlear nuclei. What do we know about them? Do they behave exactly the same way? No. Okay? No point in having two of something if they're going to do the same thing. So the dorsal projects where? Lateral lemniscus, which one? Contralateral almost exclusively. Okay? Lateral lemniscus takes it to the inferior colliculus. Inferior colliculus projects. Okay, what part of thalamus? Medial geniculate, but where else? Inferior talks to superior colliculus, and the left inferior colliculus talks to the right inferior colliculus. You do all of your auditory information, you do lots of brainstem processing for comparison between sides. You are trying to really do this without having to be conscious. You are very attuned to auditory stimuli in that sense. Okay? How about the ventral cochlear? Yeah, bilateral. Where's its first sort of standard synapse? Yeah, so the superior olivary complex, the superior olives, which get input from both, allows you to do the timing and volume differences. And where does the olive project? Lateral lemniscus to the inferior colliculus to the medial geniculate, superior colliculus, other inferior colliculus. Okay? So we've got at the inferior colliculus level, we've got lots of bilateral representation. And therefore in the medial geniculate, what do we have? Unilateral or bilateral? Bilateral. Okay? So no central lesion gives you unilateral deafness. Information is always being shared once you get past those cochlear nuclei. John? Inferior colliculus. Inferior. Yeah. Inferior. The inferiors are connected, and each is connected to the superior. Okay? Like vestibular, you do a lot of sound processing that is basically using both sides because you gain more information by using both sides. Okay? Yep. Superior colliculus. Inferior colliculus is the projection from medulla into cerebellum is the, what kind of fibers? Climbing fibers. Okay? So superior olivary complex out there in the lateral aspects of the pontine tegmentum is where you'll have that first auditory comparison. So now we've come from the inferior colliculus, and we're going to go directly right up to, so inferior colliculus through the brachium to the medial geniculate. Okay? And then medial geniculate will project to, it's auditory, posterior superior temporal gyrus, which is primary auditory cortex. Okay? So, the thalamic representation is medial geniculate. There's a primary auditory cortex. It is known as Heschel's gyrus. Okay? It's the superior surface of the posterior portion of the superior temporal gyrus. So if you look at a coronal section, and you've got your frontal cortex coming down, you've got your insula, and you've got your superior temporal gyrus here, your middle temporal gyrus, your inferior temporal gyrus, your parahippocampal gyrus, and your hippocampus here, okay? We'll talk about what's in there later, okay? This is your superior temporal gyrus. The primary auditory cortex mostly sits this way, not here. What does that mean? If you're an electrophysiologist, how are you going to get an electrode in there? It's really hard, okay? Unlike visual cortex in the monkey, which is really pretty much on the surface, here, this is pretty much hidden in primates as well, in non-human primates as well as in humans. It's surrounded by a belt of secondary auditory cortex, often called A2, and that's known as Brodmann area 42. So this is the counterpart of 17, and this is the counterpart of 18. And you've got some processing going on. So when we think about where it is, here it's drawn, it's much too big. It's actually very tiny back there. But it is, in fact, got a frequency map. That's the primary data that you have for sound. Okay? For sound, your primary data is frequency. For vision, it turns out to be orientation and boundaries. Okay. That's B is the exact mirror image of the picture I just drew down here. So this is a coronal section right through here. So when you look at the lateral aspect of the brain, here, you've got your temporal lobe and your superficial. You can't see into the insula right here, but here's your superior temporal gyrus, and then it folds in because it's completely contiguous. This is the insula. This is what we call the frontal, I don't know if I've used these words yet, the frontal operculum. It's what hangs over. And if that's the frontal operculum, what is this? Temporal operculum. And that superior surface of the superior temporal gyrus and the temporal operculum is where you find it. Okay? So you're looking deep in. This is what you would see from the lateral view on the surface, but in order to see that cortex, you have to open it up. So here it's drawn, it's actually tucked in there. And so if I were to take the coronal section that was just shown and draw it like this, and then look in, it would be the flat surface there. And it is relatively flat and wide, and you can actually see it on coronal sections, and it has the equivalent of a line of Gennari. It has a line of Ballinger, because it has a terminal field of a dense number, tightly packed, thalamic sensory fibers. Okay. So, auditory and sensory, we have really no understanding of higher levels of sensory processing. If you think about it, you go from pure tones that you use your cochlea to isolate out of the noise and out of sound, and then you recreate what is, in fact, you do the reverse Fourier transform. You are making the sound complex mixture out of those pure tones based on their power and the frequencies. Okay, so for those who did MATLAB with me, it's the exact thing we did. We took sound, we got to a power spectrum, we take the power spectrum, you go back to sound. That's what your brain is doing. We have no understanding of how it works, nor do we have any real good understanding of how you go from sounds to understanding and recognizing what units of speech and language are. There's really interesting in vivo human electrophysiology being done in the ORs to really get at what that is by having people who are having awake craniotomies with electrodes in place doing tasks as part of a research project. All right, so let's talk about somatosensory. So the nucleus, VPL and VPM, it has a somatotopic map. We've built this somatotopic map all the way from the periphery. We've built it up in basically three major fiber bundles, what would they be? Let's think. Pain and temperature, spinothalamic. Light touch, vibratory, position. Well, when they're coming up to the thalamus, they're not dorsal columns anymore. What are they? Medial lemniscus. And then there's really one more. What is it? We've got some segmental that's bringing up a little bit of gustatory stuff from solitaris, but they've got a big one that's not really covered here. So where does the spinal thalamic tract, where does it get its last neuron in? Well, remember the trigeminal nuclei they run sort of and join it. But how about the fibers that come from the principal sensory nucleus of 5? They don't merge in. So that there are separately sort of from 5 as well as 7, 9, and 10, there will be things that come in what we often call the trigeminal thalamic tract. You can sometimes see them separate from these bundles. So that's the final piece. That's the rest of your head, as well as inside the mouth and down into the viscera. Okay? Remember that somatotopic map goes all the way from the top, from your feet, all the way down in. All right? So we've got all of those, and they're organized. So we know that we have somatotopic. And we want to preserve that. But what else is going on here? We have different modalities from the same point on the body. Okay? So do we want to keep them separate? Are we going to have a vibratory thalamus, a light touch thalamus, a hot thalamus, a cold thalamus? No, you've got one map of all sensations. So in fact, these have to now mingle and build you just the somatotopic map. And that somatotopic map is going to project in a somatotopic organized fashion onto what part of cortex? It's on the screen. Postcentral gyrus, Brodmann areas 312, and it gives you what we call a homunculus. Okay? It's a little picture of a person. All right? So where are these? Well, here is, where's my, so here's VPL, and then VPM is going to be medial. And if I come back here, the somatotopic mouth puts the mouth most medial. The deeper in you go to the thalamus, closer to the midline, the more you are moving to the center portion of that map. So the most medial portion will be visceral sensation deep inside the GI tract. Okay? The most peripheral, if you have a tail, would be the tip of your tail. Okay? So, how have these fibers gotten here? You still can separate them. Yep. They are somatotopically, most thalamic neurons do not, as my understanding is, most of them do not get multimodal input. So that when you look at cortex, they are still, the modalities from a point in space are still distinct in terms of their firing patterns of what they elicit. They may share some neurons, but they do not have, they're not all composite yet. That's higher order processing. So we begin. We've got red, blue, and why do we have no green here? Well, if they're coming in the dorsal root ganglion, look where they're traveling. What kind of fibers are these? Labeled up at the top, but you could look at the anatomy of the cord. Where are they? Where are they coming in and going? Dorsal columns. Why are some of them making a synapse here? Those would be going and projecting up to the cerebellum. Remember, your proprioceptive fibers go synapse in the nucleus of Clark's column and give rise to the dorsal spinocerebellaris. So these are our dorsal columns. They come up to the dorsal column nuclei. They project across the midline and the arcuate fibers. They rise as the medial lemniscus. They've got their somatotopic map. The leg swings out lateral in the pons. They move further out lateral and join the spinothalamics. And they come up into the lateral and then more medial, lower extremity, upper extremity, portion of VPL and VPM. There's a very medialmost portion here, which is green. And those are the fibers that came for dorsal column. Where would they have come in? Dorsal column equivalents for the head? Well, what are your choices for sensory inputs into the head? Somatosensory input? 1. Let's shake heads, yes, no. No. 2? Yeah, 5. Okay, and 5 has multiple targets. So what's the target for the dorsal column equivalence? It's the principal sensory nucleus of 5, which then does what? Projects across the midline and joins, and those are the fibers that go to the head region here. Okay? Who are we looking at? What fibers? Look at them in the cord. They come in, they synapse in substantia gelatinosa, they cross anterior to the central canal, they run in the anterolateral system. These are your spinothalamics. Where do they end up? They make the same map between VPL and VPM with the same map of the body. And now where's the head going to come from? Where are the green fibers going to come? They come in from the trigeminal. Some are going here in the principal sensory, but the other ones are going where? Yeah, they run upward down in that tract of the nucleus of 5. They get to the appropriate level to make their contiguous somatotopic map, go across the midline and run as the trigeminal thalamic. I think it's called that here. Now here they draw it as being part of the medial lemniscus. And those are the green ones that come in here. They're going to be both from the principal sensory and the spinal tract of 5. Here's your somatotopic organization, easily drawn, best drawn for an animal with a tail because it lets you see the two opposite ends. And it's not really just the tongue that goes medial, but all of the visceral sensation is the most medial, okay? And the tail is by far the most lateral, all right? Makes it easy to remember. You can just see that visualization of the small little monkey. So we've now really hit the major sensory cortices. We've talked about primary somatosensory. We talked about Brodmann area 17 primary visual. We've talked about primary and secondary auditory. And then the uncus, which we'll talk about again when we do the hippocampus in about 2 weeks, is that primary olfactory. It's close to the insula where it will meet gustatory sensation, but without having gone to the thalamus. But it's a pretty small representation. You have very poor spatial localization for that. If you think about it, you have really good spatial localization on the tip of your finger. You've got lousy spatial localization on like the back of your neck and your body. And in terms of like, is it on the anterior wall or the posterior wall of my stomach? You have no ability to discriminate between them at all. All right, so where are these? I'm probably hammering this too much. Okay, Brodmann on the medial aspect, the primary auditory on the superior temporal gyrus, little bit of primary visual wraps around the occipital pole in many people, and then on the medial surface and on the lateral surface, we've got our postcentral gyrus. So this is the standard picture of the homunculus, and it comes in large part from Wilder Penfield in Montreal in the 50s, who really pioneered awake craniotomies for epilepsy. And among the things he did when people were not completely anesthetized was he stimulated the brain and saw what happened. Did you localize sensation? Did something move? And you can basically move your electrodes around and go zap, 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 and see where you get something moving. So you can build this model, and if you look at it, it basically puts the hip at the convexity, the leg draped over the midline, and then has a disproportionate representation of hand and face and mouth. Okay? And you know that. Think about trying to appreciate, trying to get something that's stuck between your teeth out with your tongue, okay? You can feel all the three-dimensional objects, the three-dimensionality of that piece of food that's stuck between your teeth, okay? Your tongue has incredible spatial resolution, as does your hand. But other parts of your body, you can't tell how big something is. You have very poor discrimination between multiple points, unlike the tip of your tongue. And that's because the somatosensory representation of it for the physical area is enormous. And the same is true for the hand and especially for the fingers. So how do we measure that in people? Okay, you can do it. You guys talk about bold in the imaging course? No, all right. So MR is essentially a magnetic measurement. Blood contains hemoglobin. Hemoglobin exists in two states, with oxygen and without, and they have distinct magnetic properties. So by looking at the magnetic signal out of blood, you can tell and make an estimate of how oxygenated versus unoxygenated it is. So that's the first premise. You can make that measurement. The second is that when brain is active, we'll talk about this late in the course, when brain is metabolically active, which is what happens when neurons are firing, so active areas, they actually summon up increased blood flow. So there is increased perfusion in the setting of local activity. So you will then, if you have increased perfusion, what do you get? More oxygen or less oxygen? More oxygen. And the reason you get more is that you are using more, that's what the metabolic demand is doing, but you're in normal circumstances, and this is the first caveat, the vasculature has to be normal. In normal circumstances, the level of response of increased vascular flow is greater than the need. So even though you are more active, the local oxygen, oxyhemoglobin goes up, okay? Rather than high demand making it go down, you overcompensate with the vascular change. And you can make that measurement, okay? Measurement was first made actually at the Martino Center in about 1990, but it's now used as a really widely made method for looking for function. And it's based on a whole lot of, as I just said, multi-step assumptions. Normal vasculature, normal autoregulation, so that demand of neuronal activity leads to change in vasculature, and that your measurement reflects the level of the activity. It means, though, that you can do things like this, which is you can ask somebody to do a simple movement while they're in the scanner and make the measurement. And what you do essentially is you measure the oxy-deoxyhemoglobin ratio in an area of brain when their hand is still, and then you say, okay, now move your thumb for 30 seconds, and you measure it during that time, and you say, wait, hold your hand still, you measure it for 30 seconds, and you do that multiple times. So it's almost always done with multiple trials so that you get high enough signal to noise to be able to see what is essentially about a 1 or 2% signal change, okay? So these are all based on very small differences that require repetitive going through testing, okay? And you can do that, and you can localize it, and you can build models of what function lies where. It's probably pretty accurate for simple sensory and motor output. The interpretation of it in complex behavioral tasks, I think, is far more complicated because you're making lots of inferences about what's going on, and it gets harder and harder to do the averaging in a reasonable way. But if you think about it, the brain's a huge place. Here you can do it because you know exactly where to look. But what if you don't know where to look? What are you going to do? Well, you're going to look everywhere. You're going to divide the brain up into tiny little voxels, small volumes, and make lots of comparisons. And this is where the cautionary note comes, which is you make lots of comparisons, you need to be really good about your statistics, okay? So here's a wonderful poster. This is, I mean, everybody who works in this area knows it. The poster is neurocorrelates of interspecies perspective, and it's a argument for multiple comparison corrections. And here's the salmon in the scanner. And after doing the testing, here is the voxel that lights up at some high level p-value 10 to the minus third. And then when you read the methods, subject, 1 mature Atlantic salmon, salmon salar, participated in the fMRI study. They don't comment about IRB approval. Salmon was approximately 18 inches long, weighed 3.8 pounds, and was not alive at the time of scanning. Okay? This is a dead fish put in the scanner. If you do the statistics poorly, you can find signal someplace. How come? Well, even in a salmon brain, which is pretty small, you're making thousands of comparisons. And you're looking for small differences. The noise is high. So you need to really either have underlying good neuroanatomy and prior knowledge to direct you to targets that you look at. But sort of big global searches, you have to have very robust signals if you want to have good statistical confidence. So, let's talk about association areas. Yep. Yep. Well, they usually, so the issue is it depends how you make the measurement. If you do a global comparison and they're thinking about something else, you will find they're doing visual imagination about something else, they're listening to music while they're doing it, you will get lots of other signal. If you say that they're in there, they've got earplugs and headphones on, they get the instructions, we're going to ask you to move your hand when the red light comes on, close your eyes and move your hands. So you diminish visual stimuli, you diminish auditory stimuli, you have a specific task, and you look at the area that you suspect, then it's very robust. Yeah. Right. So you're always comparing stimulus or activity or stimulus on and activity stimulus off. So you always are doing that. And then if you're doing cognitive testing, if you're doing say a word reading task, you will be giving them, and you're looking for some aspect of it, you'll give them lots of words. Some trials are the task you're actually asking them to do, and others are either a distractor or a false task, and what you're looking for are differences. So you always have to do them as comparatives. No, all you're doing is measuring the change in blood flow in response to activity. And so it depends on how much activity you have, and there's no reason to assume that one of them requires more than the other. So it's a multi-step away from reality measurement. It's useful, but I think you have to always be very careful about them. All right, so we talked about our unimodal association areas, how you go from first to second, and then to third, and so on. But at some point, you want to bring these together, and that's where you come to unimodal, and we have two basic huge areas, one of which is sort of this vast untapped zone of the parieto-temporal-occipital region, mostly out laterally, okay? And it's going to have convergence of really all three primary sensory modalities. Somatosensory, coming from the posterior central gyrus and the front anterior boundary of the parietal lobe. Vision, coming from the medial occipital surface and spreading out along that as you process it. And then auditory from the posterior surface of the temporal lobe coming in. And so you come into this and you're going to have issues about language. That's how you know what you have heard. So the first thing to do is to realize that reading and writing are not the intrinsic components of language. Language essentially is oral. You hear it and you speak it, okay? That's what is evolutionarily driven. Written and read language is a much later acquisition. It co-opts and uses pathways that depend on the intrinsic language circuitry. But when we talk about language, we're talking about listening, having somebody listen to you, and you listening to them speak, okay? Talks about spatial performance. Where are you in space? Do you know where you are? Can you take your image of space and use it to remember where you were? Do you get lost? Are you able to find your way? And then a whole bunch of other things that you need. So if I ask you to do a complex motor task that involves both motor planning, but knowing something about sort of what you're doing, the typical thing is, well, not for me, but I could ask many of you to show me how you would comb your hair, okay? And you would do something like this, okay? But the ability to make that movement requires understanding the instruction, understanding what combing your hair means, and then eventually putting together that motor output. And the failure to be, you have normal strength, no ataxia, but you can't do that task. That deficit is known as apraxia. You can't do sort of practical activities. Okay? And then more anteriorly, in front of our output, we're going to have the decision-making about what to do based on everything we have here. Decision-making, planning, and executive function, which is the global term for things that we think about as judgment, foresight, planning, making decisions, balancing competing needs. So what do we have? We have primary cortices that sit below secondary cortices that sit hierarchically below association cortices that come from sensory receptors through distinct thalamic nuclei. So we have multiple parallel channels, and then finally, at higher cortical levels, we bring them together. We've strenuously kept these apart. We've even kept different sensory modalities from the same part of the hand separate, okay? All of those fiber bundles were, kept apart. I mean, life would be much easier if everything ran into, X thalamic tract and it was all a jumble and you just sorted it out later. But we don't do that. We keep all of these as incredibly parallel structures and parallel processing streams. And we, by extension, use what we know about this one to make assumptions about these. All right. So let's switch over and think about motor. So here we've got more anterior thalamic nuclei. Does that make sense? Yeah, the motor strip is in front of the sensory strip. You'd expect these to be right in front of VPL and VPM, and they are. They are VA and VL, and VA is really more A than V, okay? But it is still called VA-VL. And what are the major inputs? Well, we know they're going to talk to cortex. We already know that this is the thalamic target because this is the motor output for cerebellum. And then basal ganglia that we'll see in about a week and a half is also going to feed in. Okay? And where do they go? Well, they go, in fact, to all of those motor areas. So that's by all of the cortical motor areas. That's not just Brodmann area 4. What's Brodmann area 4? Yeah, that's the final motor cortex, output cortex. Cells in there, the big pyramidal cells that sit in layer 5, those are the BET cells, which are the upper motor neurons, and their axons are the corticospinal tracts. Okay? So they do project here, but it also projects to what we often call the supplemental motor area and premotor cortex. Those are the components that are involved in the planning. Okay? How is it that you're going to make the motion by generating a sum of anterior horn cell activity? Okay? That anterior horn cell is a simple, that's the simplest, one cell talks to another that talks to a muscle, to a motor unit. Okay? But that complex movement is a trajectory made of multiple changing patterns. And the patterns that you're going to generate from this as your output device come from these. Remember, you come from the most complex down to your output of the most simple. When we think about the VA-VL, depending on which book you read, and this is the way I teach it because it's based on the book I learned it from and the way I was taught it, is that for the most part, the cerebellum, which has the L's in it and no A's, goes to ventrolateral. And the basal ganglia, which, yes, it does have 2 L's in it, but it also has 4 A's, okay, goes to ventral anterior. Okay? So you can either use the A's or you can try and use L's sometimes and not the other time, whatever you want. But they are relatively separate targets in thalamus that next coincident project to cortex. So that you don't have a separate target of VA and VL, but they come together. So the convergence, in sensory modalities, we were trying to get the convergence. We scrambled them up in the sensory thalamus, we scrambled them up by modality. But here we're keeping the type of information anatomically separate still. and projecting it onto the thalamus. Okay? So VA and VL, and they're going to project to the motor and the premotor areas here. Now remember I said we have this bizarrely termed structure that sits in front of the motor components that even though it's in the frontal lobe, we call prefrontal. Okay? That's the way it goes. So, I've got a little bit more color coding here. So when we think about that, we've got our primary motor and premotor here in purple, the dark purple, and the light purple both project in there. They've got the dark purple coming in as dots to the medium, more to the more supplemental, but they both do project to both. Okay? So VA, VL to that premotor planning and motor component. And then remember, it wraps around on the medial surface. So what type of anatomic function, what anatomic areas would you expect to be here on the medial surface? Face, arm, hand, leg. Leg, exactly. The same homunculus maps for somatosensory over here on the posterior and motor on the pre.
The homunculus I've never seen, the tongue and more inside stuff mostly. So, I don't know, I get that confused. The leg's not.
Leg is on the lateral aspect.
Oh, on the lateral aspect?
Yeah. Leg is on the leg is on the medial. The face is down here, out here on the inferior boundary of the lateral aspect. All right, so what do we have to do movement? We've got the output, the precentral gyrus. We've got inputs into it from premotor cortex, and then the planning of what you're going to do. This is the intention, this is the organization, and this is the actual output. Okay? So you mostly go 6 anterior to 6 posterior to 4. And that's really based on what areas become electrically active as you give an animal the task to do. What fires before the lower motor neurons fire in Brodmann area 4. And you can do it both by imaging and by electrophysiology. In people, you mostly do it by imaging, although you can do it by electrophysiology with awake craniotomies. So where are they? Here they are. And if you think about that homunculus, the hip sits up at the convexity here. Your thorax, arm, there's usually a little knob here, an omega, bend in it, which is called the hand knob, and then your face below. Say a few words about eye movements, and then we'll take the break. So how do you move your eyes? Well, we know the mechanics in the brainstem. And we know some stimuli that get you to move your eyes. We know that turning your head drives your eye movements by the vestibulo-ocular reflex, right? You stay looking at an object, if you're awake, while your head moves or your body moves, okay? We know that you move your eyes in response to a short visual stimulus through the colliculus, the retinotectal projection that feeds down. But what is it that does when you decide you want to look at something? Where do you make decisions? Make decisions in cortex. And a decision like that is made in premotor, is made in the prefrontal cortex. You're doing that. And it's a movement like anything else, like deciding you want to, move your hand. Yet there are distinct areas that move the eyes. There are the so-called frontal eye fields that are involved in voluntary conjugate gaze. And the key here is the left frontal eye field. What part of space do you expect it to be looking at? Right. Okay? Your left hemisphere cares about the right side of the world. So those eyes are in charge of exploring the right side of the world. If you have a lesion in your frontal eye field, what will happen? Okay? Normally my left side of my hemisphere is making my eyes explore the right side. My right side is making my eyes explore the left side. I take my left hemisphere away with a lesion. Where do my eyes end up going? They go to the left, and they don't typically cross the midline. Why would they do that? What's controlling them? The intact right hemisphere. And that's what's meant by people saying the eyes look to the lesion, okay? That frontal lobe, frontal eye field lesion, when you lose those eye fields, your eyes will only be driven by the contralateral side, and they will deviate towards the side that's missing, okay? You also have, just wait, you also have occipital eye fields. Why would you have them? Or parieto-occipital eye fields. Why do you have them? What do you do with, what information do you have in the occipital lobe? You've got your vision. You've got the awareness of where objects are in space and how they're moving. If you want to track an object, what do you need to use? Its immediate prior position, okay? So you may have the idea you'd like to do it, but the data you need to do it is actually in the parieto-occipital region. So those eye fields are going to drive it. So if you think about it, if you have a volitional eye movement, like a saccade, where you're surveying an object, your frontal eye fields project down. Why do they go to the superior colliculus? What's the beauty of the superior colliculus? Yeah, it's got that spatial map that lets you go, oh, I want to be over in this new coordinate. That feeds into the horizontal and vertical gaze centers. What do they get information from as well? Vestibular and into your ocular motor system. Okay? So now you've actually gone not just the circuitry in the brainstem, but you've actually driven it from above. Okay? If you're trying to smoothly pursue an object, you have to both want to, but you also need to have the direct visual information. And here, this is the parietal eye field. Sometimes it's called parietal. Sometimes it's called occipital. And that comes into the pons for the most part. Why? You're making a smooth movement. You use your cerebellum. So you go from your basis pontus into the cerebellum, and you generate that coordinate that says, here's how you make the smooth movement. You bring it in with your vestibular information, and you do all of that. All right. Let's stop, pick up again at 1:15, and we'll talk about speech briefly. We'll talk about speech a lot, language a lot more on Wednesday, and then we will finish with the non-sensory motor thalamus. All right, so the other area that I want to mention today, but we will spend much more time talking about on Wednesday of next week, is Broca's area. So this is the premotor and motor planning area associated specifically with speech, okay? So when you think about speech, it is an incredibly complex motor activity. It requires my regulation of mouth, requires regulation of tongue, requires regulation of upper airway space, and it requires my control of respiration. All of those things are happening. But in order to do that, not just for a simple motor activity, but to use that to put language out, there is a distinct additional premotor planning area that does it. So that someone who has a lesion, a small lesion in Broca's area, which is in the inferior frontal gyrus. So let me draw a picture. Let's draw the picture here. So here is our brain. Here's our central sulcus. Okay. Here's our superior, inferior, middle and inferior frontal gyri. The opercular part of the inferior frontal gyrus. Where's the operculum? What does the operculum hide? The insulus. So it's going to be down here. So down here, if this is my precentral and postcentral, right down here is where I will find Broca's area. And that is more than just the mechanics of speech that are required, but rather the content of language, the ability to get the language out. So somebody with Broca's aphasia, with a lesion there, can't get their speech out. Their control of their respiratory system is fine. Their tongue movements, if it's just in Broca's area, will be fine. Their ability to shape and move their tongue and lips will be fine. but they won't be able to get language out. Language is different than just the motor mechanics of it, and we'll spend a lot of time on Wednesday talking about it. All right, so there's Broca's area now here in the dotted line over on the side. All right, so let's go to the ones that are vaguer, okay? Motor, somatosensory, auditory, gustatory, those are all pretty straightforward to think about what they do. Let's talk about the fuzzies, all right? So we're going to talk about a couple of them, and we're going to look at the color-coded map. So we're going to look here. So we've got the anterior nucleus. What's the anatomic landmark where the anterior nucleus sits? Visualize not the thalamus as this oblate football sitting free in space, but in a nice cross-section. So it's going to be the foramen of Monroe between the lateral ventricle. Which side would the lateral ventricle be on? So the lateral ventricle sits here. Foramen of Monroe sits here. And what's here on the medial surface? Third ventricle. All right. So this is sitting right at the foramen of Monroe, bulging forward, and it is going to project this. What is that loop? Cingulate gyrus, that limbic loop, that edge. As you think about that cortical sheet, it's completely contiguous all the way around until you come over here. I have no idea what that is. Absolutely no idea. All right. It's not on my screen, so I assume it's something in the room. Who knows? All right. So here we are. And so that's our projection from the anterior nucleus to the cingulate gyrus. So here again, we've got that anterior nucleus. Where is it? Here it is. Is this a left or a right thalamus? This is tricky. It's a left thalamus, but it's angled the other way. Okay? Here the left thalamus has been shown with its true orientation, but here the slices have been slid in the other direction. Not intentionally, but it looks more confusing. But here it is, and this would be, again, we'd go from the lateral ventricle on this side through the foramen of Monroe to the medial. We've got our anterior nucleus on the anterior and medial, and its major input is going to come from a structure known as the mammillary body that we will meet in the final week of the course. Okay? So we have one major input and one cortical output. It seems life is so simple. Okay? Well, where is the mammillary body? I will show you. It is right here. We will talk about it when we talk about hypothalamus. And where's the thalamus in this picture? Somebody tell me which way to move my pointer from this spot. Medial, lateral, anterior, superior? Medial. I can't move medial. I'm on the midline. Anterior, tell me when to stop. Okay, now what? Caudal. Caudal's an interesting kind. We don't use caudal when we talk about cortex. Yeah, I would say either ventral or inferior. Yeah, so here, we're right in there. We can see it. That's the foramen of Monroe right here. So the anterior nucleus of the thalamus must be right here. So in fact, our mammillothalamic tract is going to run just like that, slightly deep in the block. Okay? And then the anterior nucleus of the thalamus is going to project to the cingulate gyrus, and it projects all the way around the cingulate gyrus just like that. Okay? We'll talk about its role and function later. But what I'm going to show you eventually is that the mammillothalamic tract, which is here, mammillothalamic, from the mammillary body to the anterior nucleus of the thalamus. That then projects to the cingulate. The cingulate, we will see, projects to structures that get us into the hippocampus, and the fibers that leave the hippocampus will come back to the mammillary body. So we will make a loop that will go 4 synapses and close. It will also have branches at every point. There's no point in making a closed loop in the nervous system if all it does is reverberate on itself. It has to have points where it intersects with other circuits, but this is a well-recognized loop known as PAPE circuit that we'll talk about in 2 weeks. All right, we'll skip that for now. We'll come back to it later. All right, so lateral dorsal. Okay, remember that lateral dorsal sits behind the anterior and actually really right on the dorsal surface. Okay, the only problem is it's not really lateral. Okay, so it's one where the dorsal makes more sense than the lateral. Its major input is from the hippocampus, and it too talks to the cingulate. So we've got two things that are going to cingulate that have different types of information that have in fact come from the hippocampus. Okay. They are connected to hippocampus and related structures, and as we'll see when we get to the hippocampus, hippocampus is critical for memory, but so are the output structures that it uses. If you have information in hippocampus, you use it to process memory, but hippocampus is not where memory stays in the long run. In the long run, it is somewhere in cortex. Frankly, nobody knows where, okay? But it's in cortex. How do you get things to cortex? You go through the thalamus. So damage to these thalamic structures lead to inability to process memory, okay? And in particular, these plus dorsomedial nucleus, because they sit up against the ventricle, are susceptible to injury in Wernicke's, which is, anybody remember this from biochem? Rick gives the vitamin lecture. Oh, it's not B12. B12 is subacute combined. It's thiamine. So somebody comes in, they have acute memory disturbance, they've got eye movement abnormalities and gait abnormalities, they need to get treated with thiamine. Okay. Next. I don't even know what I'm trying to highlight here. Let me just... Oh, okay, dorsal medial. So dorsal medial, we're working on frontal and prefrontal. So dorsal medial is not as far anterior as the anterior, but it is relatively anterior and medial, and it is going to project to the entire, both dorsolateral prefrontal cortex, medial prefrontal cortex, and down on the inferior surface, the orbital frontal cortex. Okay? It's a pretty big nucleus. It runs about half the length of the thalamus, is widely projecting to those regions. What is it involved in? Or what are we doing up there? Yeah, you're making decisions. You're not processing primary information. You're not doing your motor output. You're doing the thing that sits in between those two. That is where you are, in fact, doing thinking. Okay? So dominant input and outputs are prefrontal, but some come from other areas. So you do get information about your visual field, and you do get information from your medial temporal lobe, including hippocampus, that tell you about where you think you are and context. Because while you may, be in this room, the context in which you are in this room can be different. So it's not just where am I, but where am I and what is the context of it? What do I remember about what I'm supposed to be doing? All right. When we think about those frontal areas, the prefrontal, lots of it is made up of superior, middle, and inferior. When we talk about prefrontal, we sort of would draw a line like this. We will have out here the so-called dorsolateral prefrontal cortex, DLPFC. It's a common abbreviation that you will see in books. And then on the orbital surface, we have the gyrus rectus that sits right beneath the olfactory bulb and the so-called orbital gyri. And now let's move to the back, to the pulvinar. Okay? It's big, it does have structure to it, and we don't worry about teaching it to you. Okay? That's a freebie. You just have to know pulvinar. You do not have to know any of the substructure to the pulvinar. Its inputs and outputs, it's the counterpart of the dorsomedial, which is the prefrontal. This is with parietal lobe. This is the parietal, your somatosensory, auditory, visual, integrated world. That's what's going on here. Okay? It's got information for processing visual and other integrated sensory data. Some of it, there's a portion that is relatively unique to primates, and there's a lot that's evolved in visual attention. Okay? You spend a whole lot of your time trying to figure out where to look, because something bad might be about to happen. Okay? So that's really what you're doing. I mean, it's not that you're trying to be able to, look around at what a lovely day it is outside. You're continually looking for something bad about to happen to you. And you spend a whole lot of time paying attention to that, because if you don't, if you don't have a good medial pulvinar, evolutionarily, you're going to be a dead end. Okay? You need to have that working in order to be able to transmit your neuroanatomic both structure and knowledge. So where are these projecting? Well, they're going to those vague structures in the parietal lobe. We'll talk more about deficits on Wednesday. But on the lateral aspect, there'll be what's color-coded here in green, reddish, and blue. Superior parietal lobule, the supramarginal and angular gyrus together make up the inferior parietal lobule, IPL, and then the precuneus. Why not the purple stuff here? What's that? Somatosensory. That's not higher order processing yet. Pulvinar really talks to higher order processing. So, where is it going to have to go? Well, it's in this. Here it's drawn very small, but if I were to draw it, I would have drawn the boundaries more like so. Okay? So that's where you've brought this information, this information, this information that's not even highlighted here together. Those came from VPL, VPM, lateral geniculate, medial geniculate. And then at the next point, you're going to have pulvinar connections where you additionally have thalamic inputs in there. So here are recordings from primates. This is a rhesus monkey being recorded from in the pulvinar. And if you look, there are certain patterns of stimuli for which the pulvinar just goes berserk. Okay? So these are, the way these are presented, they're raster plots, so they're basically the amount of a number of axons firing, or neurons firing, spike density, relative to time 0, which is when the image is shown. So something that responds to the image will have a burst. And something that is completely agnostic to the image, the frequencing rate won't change between the left side and the right side of that vertical line. And so what do they respond to? Snakes. Is that something good to respond to? Yeah. You are paying attention. You, as you walk around, are in fact having your pulvinar continually worry about snakes. Okay? It really is. And you may are not aware of it, but your pulvinar is wired for the object that looks like a linear coiled something nearby. Okay? And that's very different than the shape of a face, the shape of a hand, objects, or other things. You are tuned to things that are important. All right. So, yeah. Well, so there's recognition of all sorts of different types of things, but the ones that you are most driven by are in fact things that are threat. Okay. So we've talked really, if I step back for a sec, we've talked about the sort of, relay folks. And they're mostly sensory, but they do include motor. And they really bring information from a non-cortical source to a cortical unimodal device. Either the first processing step for sensory or the organization to get yourself organized for motor output. We talked about the association ones. Lateral dorsal, anterior, dorsomedial, and pulvinar that go to associational cortical structures. Cingulate really functions that way. And they are involved in sort of providing the sort of continual feedback for those as they balance and try and make decisions. Okay? So those are the two big categories. They're different, but now we're going to come to the third. And these are the least well understood because they are the least defined anatomically. They are hard to see, And they are also widespread. And the problem with things that are widespread is that most lesions don't take them out selectively. Okay? So you can have somebody who loses their pulvinar based on a vascular territory, but you're not going to have someone who loses just the reticular nuclei based on a vascular territory, because they are in between two other structures that they share with. So you're going to see deficits that are associated with multiple structures, not just the one that you're interested in. So if you really want to take them out and understand their function, you have to do either genetic manipulations to ablate them, genetic manipulations to make them sensitive to drugs, or genetic manipulations to make them sensitive to light and then deliver light and turn them on and off. And you can now say, what do they actually do? Or you can take animals and you can shove a whole lot of electrodes in and say, and record from them and say, what is the context in which they fire? And if I depolarize that region by overdriving it and I take them out of the circuit, what do I see as a behavioral consequence? Those are really hard experiments to do, and anything that's done in a non-human primate is just an enormous undertaking. Those people who work on people who do PhDs with non-human primates, might want to guess how many animals they have for their thesis? Yeah, 1 to 2. And the first year to 2 years is spent doing what? Training the monkey, okay? This is an enormous investment. It is incredibly hard, and they are incredibly, I mean, they bring an enormous amount of power to our understanding, but you are not going to do the same thing as, 10,000 Drosophila or, 50 to 100 mice. You just can't. So initially they were, but realistically, if all you can do as a neuroscientist is cut brains, stain sections, and name things, that's what you do. And then once you've named them, and you now know how to trace pathways by basically injecting dye into different brain regions, either as beads or as horseradish peroxidase, a couple of things, you inject them into every structure you can, and you see where they go, and you discover that they're wired differently. And some of them, in fact, are interneuron circuits that regulate intrinsic thalamic function and contribute, in fact, to disease through that. But basically, all of anatomy is done because it's what you could do. Pathway tracing and defining fiber connections is what you could do. And then you move on to function when you finally have the tools to be able to do that. All right. So when we think about these, and in fact, this leads right into PK's question, many of the reticular nuclei sit as inhibitory interneurons that either inhibit each other or inhibit the thalamic nuclei, so that they are involved to a large extent in coordinating thalamic activity, so that not any stimulus in every area is running at the same time. You're essentially gating channels of information so that they are alternating what comes to attention. And the other thing that you need to know is that many of these neurons have distinct subtypes of channels, including the T-type channels, calcium channels, and that alterations in them can lead to disruption in thalamic function. And you can basically do a global manipulation of them because these are just about the only neurons that have them. All right. So when we think about them, they're located in the white matter bundles that separate the other nuclei. And there are also ones that sit sort of pretty close to the midline as this thin sheet right along the edge. We break them into 3 zones going from medial to lateral, and they're slightly different in how they are connected. The midline ones, don't worry about ever seeing them. But the midline group, okay, get input from nuclei in the brainstem that are cholinergic and serotonergic. I mentioned the serotonergic nuclei early on when we were talking about the brainstem, second lecture. They are the raphe, okay? These are the neurons that are the target of drugs like Prozac, which are selective serotonin reuptake inhibitors, which will do what? They will selectively block serotonin reuptake. Where? at the synapse. So the consequence is you enhance serotonergic transmission. Okay? Very simple. And they get major inputs from there, and they output to a whole bunch of structures that are really associated with both memory and emotional valence that then filter and superimpose on your prefrontal decision-making process. Am I feeling good? Am I feeling bad? And this is the gating input for that. We come to the middle range, and we've got those central ones that are intralaminar. Okay? Centrolateral and paracentral, don't worry about the names. They get inputs from just about everywhere, but they go back to what type of cortices? Prefrontal and parietal? Multimodal. Okay? Not with a significant projection to the primary motor or sensory cortices. And then finally, the so-called intralaminar ones. Centra median is the only one you need to know the name of. Okay? It has basal ganglia inputs that we'll talk about in about a week. And it goes out back to basal ganglia and to sort of motor cortex for planning. The only thing you need to know about this is that it is probably still thought, although the data is less clear than it once was thought to be, that abnormalities in activity in the centra median are at least in part responsible for one specific type of epilepsy. Did you guys hear from Elizabeth Thiel in the first block, or is she at the end of the course? She's at the end of the course now. So when we think about epilepsy, epilepsy is a disease of inappropriate electrical activity in the brain. Most of the time we think about it as seizures, focal motor activity or behavioral change and the like. And those are all forms of different forms of primary epilepsy. The other typical type of epilepsy, which is more characteristically seen in children, is a disorder sometimes known as petit mal seizures or absence seizures, where you will be talking with somebody, you'll be watching them, and for a short period, 5, 10, 20 seconds, they will stop interacting, stop responding, and then pick up again just where they were. Okay? So they have these absence episodes. They are absent from the room, effectively, for these short intervals. And some of that, the early models, and you can elicit similar behaviors by altering activity of centra median. And in a sense, if it's responsible for gating all of your motor output, all of the motor stuff closes down for a moment and then picks up again. But meanwhile, all the sensory processing, all the other things have been going on so that, they come right back into the conversation as though they had not been missing. Okay? Don?
Just for the absence seizures, they just blank, or do they fall on your
No, they stay blank. They do not fall. Their intrinsic motor stuff, which is all mostly vestibulospinal and all of that, stays fine. We'll talk about people who collapse. We talk about hypothalamus, and I think we'll hear a lecture from Tom Scammell, the tail end of the course, about narcolepsy. Emery? Nope. Nope. What? They skip time. Right, they're aware based on context, but if they're just sitting there not interacting with somebody, they're unaware that they're having spells. Especially if they're very short. If they're very long, then it becomes more obvious. But if they're very short, which is still incredibly disruptive to normal function, they are not aware. I mean, if they're sitting next to you and you leave the room, they'll realize. Come on, Deb. So they tend to freeze. Right.
So what you'll see them do is like you're talking to them, and then like they'll literally start staring, and then like maybe they'll eye flutter, and then they'll like kind of like almost look like they're gonna like fall back a little, but it's just like so brief.
It's so brief. Yeah, the episodes are very, very short, and the recovery. But when they recover very quickly back to full function, as opposed to people with grand mal seizures with diffuse motor activity widespread. They have a post-ictal period during which they are not back to normal that can range from few minutes to hours. Whereas for any one of these absence episodes, the recovery is very, very quick. Absence. Absence, but present absence. Petit mal. Little disease seizures, as opposed to grand mal. All right. So what's going on here in terms of thalamic function in cortex? So the answer is that in order for your cortex to be able to handle the information it gets, you need to coordinate the thalamic inputs. And that coordination of them is cortex in sort of ready state versus non-ready state. And our cortical areas that are going to be connected to each other, both in ready mode versus quiet mode. Okay? That ability to have connection from one area to another, to have that function, requires the thalamus getting both of them aligned in some way. So that the organization of cortical activity critically depends on organization and synchrony in the thalamus. And those intralaminar nuclei that provide the local inhibitory interneurons are important for that organization of thalamic activity that is essentially priming cortex to have something happen in one place and know where to send it and have the recipient be appropriately poised to get it. Okay. So, you should now, which thalamus? Yes, this is the only picture I could find that did a right thalamus. Okay? But you should be able to basically go and follow and draw out for any of these targets where those cortical areas are. Think about what the functions are. Think a bit about what the inputs into these thalamic structures would be. And that input could begin all the way down out in the periphery with a sensory receptor, or it could be in cortex. Okay? All of those. You should basically be able to put that together. So I'm going to close this morning by asking, does anybody in this room know who Karen Ann Quinlan was? So Karen Ann Quinlan is a young woman who was in a motor vehicle accident, and she was actually the focal point of the very first legal arguments about right to withdraw care. Okay? So she was in her 20s. She was in a motor vehicle accident. She was comatose from the point of injury onwards. And there was a whole long legal debate. And eventually it was agreed that they could withdraw ventilatory support. And it turned out that she actually didn't need ventilatory support. And she continued to breathe on her own for about another decade in her comatose state. Okay? At the time that she finally died, the brain was removed. It was held by a medical examiner for a quite long time. And then eventually it came to my colleague Hannah Kinney at Children's Hospital. And Hannah did the full neuropathologic examinations. You have to remember, this paper is from '94. I think Quinlan died in about 88 or 89, and the accident was in the mid-70s. Okay? So at that point, there's no useful imaging. We don't have MR. We don't know where her lesions are. And so the question was, she's been completely comatose all this time. Where's the lesion that did that? And in fact, I don't have the pictures from it, but I have the citation here. Anybody who wants to can go pull the paper. The bulk of her damage was thalamic. And why would that make you comatose? Nothing is getting to your cortex. Nothing is coming from your cortex out effectively. Okay? Your brainstem is still running. You've got your control of blood pressure. You've got your control of oxygenation. You can control your heart rate. You control your respiratory rate. That all happens down with cranial nerves, 9 and 10 and out through the phrenic, and it just happens down there. But without the thalamus, even intact cortex will not keep you conscious. Why don't we stop and then I can answer questions. All right. I actually finished ahead of schedule for a change. Monday is a holiday. You are welcome to come here and sit, but there will be no entertainment.